Thank you for inviting me. And, since I have to follow Peter and Chris, I think I don't have the ambition of telling you something new that you haven't heard from the two of them, because I think if I did, it probably would be incorrect. So I'll just try not to embarrass myself and describe the work we're doing on psoriasis. And so, I guess I like to just start from first principles, I guess with this goal of not saying things that are unexpected and wrong when I follow on from people who are so much smarter. But so, we all know that the human genetic studies work on this basic idea that human chromosomes are mostly the same. There's a few differences between them. There's a lot of differences that are rare, a few that are common. Overall, we expect that the common ones usually have very small consequences, and that some of the rarer ones can have more severe consequences. We'd like to study these variants to understand human disease, in part to enable new treatments, in part so that maybe we can predict disease and help patients make early decisions and undertake prevention earlier on. And when we take this to practice, genetic studies have been quite successful. They've been quite successful, especially in the last few years, as we've been able to genotype common variants on a large scale. Here's a list of loci identified for psoriasis between 1976, when association was identified, the MHC, to the CW6 gene, and then early 2012. And I think if you were going to draw it an update to this graph as of now, the number of loci would have doubled again. So it's quite clear that we can make progress in identifying genetic signals, but the goal is not just to identify signals, it's to answer those two questions, is to understand the biology, and some of these signals have very clear biology behind them. So for example, when you look at IL-23 or IL-12B, some of the immune messengers on this list, we've got some very good ideas of what they're telling us about the biology, but there's several other loci on this list where, I would guess that very few people have any clue, as to exactly how they're connected, and so it's a big challenge moving from just identifying signals to actually tackling those two big questions, understanding the biology and making predictions. The current challenges, I guess, are that we're now very good at doing surveys of common variation, and that as sample sizes increase, we'll identify even more disease susceptibility loci. But for some of these loci, the biological implications are not so clear, and trying to figure out how to move forward to function is challenging. And so, in the context of psoriasis and many other complex traits, what we'd like to do is to figure out, how can we make it easier to move from these association signals into biology. One way to think about doing this is to study variants that have very clear functional consequence. We could imagine doing this in the mouse, and I think increasingly there's this hope that one could do this in humans. Any gene of interest is likely to be severely defective in at least a few individuals. And sequencing allows us to identify carriers of these knockout alleles. So basically provides an alternative to doing animal or in vitro studies that work sometimes but work less well other times, because humans can be quite unique. One challenge is that because these severe defects in a gene are so rare, it's quite challenging to make these experiments work through sequencing. It's quite hard to get to the required sample sizes. And our take is that basically sequencing is only part of the solution. Current sequencing studies are usually well powered to discover variants to contribute to disease, but they most often won't have sufficient power to really link those interesting variants to a phenotype, and that much larger numbers of individuals will need to be examined to establish the effect of a variant than just to discover it. And so we think that genotyping will provide a practical way to really get to the required sample sizes and power both because they'll enable the most interesting variants to be genotyped in tens of thousands of samples, and because through imputation we can impute even additional variants that we can't genotype directly in these very large sample sizes. Kind of an example of the challenge, from our work is a recent study we did on macular degeneration. We sequenced about 3,000 individuals looking for rare macular degeneration risk variants. We found some interesting variants, complement 3 and complement factor H gene. But if you look just at the sequence sample, our top signals had p-values of something like 2 times 10 to the minus 4, which in the context of thousands of variants that come out of these sequencing studies is not quite sufficient. And only by being able to extend the sample size for genotyping could we really convince ourselves that these are true signals and that they stand out. So for example, when we went from about 4,000 individuals with sequence data to about 12,000 individuals that we could genotype, the signal went from 2 times 10 to the minus 4 to about 1 times 10 to the minus 9. And with additional confirmation in other studies, it's not quite clear that it's right. Okay, so in psoriasis, what have we been doing? So we started by identifying a set of high priority regions. There's about 100 of them. They include about 769 genes, a mixture of genome-wide associations studied at Loci, genes in pathways that seem connected to psoriasis either through genome-wide association or previous functional studies, loci implicated in other autoimmune disorders. We sequenced about 5,000 individuals working with Baylor and ended up identifying a set of about 160,000 variants. 120,000 of these were newly discovered. Most of those were actually just seen in a single individual, and about 20,000 of these variants alter protein sequence. This is the frequency spectrum, just to make the point that in this set of 5,000, most of them are very, very rare. And if you do an association analysis based on the sequence data, you see interesting things. I think in this particular graph, almost all the signals, except that red one, that reached significance were known. But it's clear that if we want to really decide, which of these 20,000 protein changing variants, for example, are the most interesting, we need to reach a larger sample. And so we set out to do a second part to this experiment, which was to genotype the most interesting variants in an additional 11,000 individuals. So we took the most interesting variants from the sequencing experiment. We wanted to be able to impute other interesting variants as they're discovered in different studies. So we wanted to have a GWAS backbone and coding variants from various studies. The whole experiment was carried out over a period of about 4 months, starting from designing the array to delivering the DNA samples to getting the genotypes back. I'm going to show you a little preview of results at the end, although to be fair, we've only had the data for a very short while. We haven't really digested as fully what it all means. So the first step here was actually just preparing to design the array. We generated a list of potential custom content that was interesting to us, about 260,000 variants. We delivered this to the Affymetrix design team, and they gave us back a summary of their thoughts on each of the variants. Some variants were already on the Axiom core chip. Other variants were not on the chip, but they had a series of probes that they had previously tested, so they thought, we know how these will work. Other variants, they said, we think these can be tagged, even though they're not on the chip, and we don't have a validated probe. There was a small set of variants that they said, we don't think these will work. And then there was quite a large set that was completely new to Affymetrix, and they said, we can design de novo genotyping probes for these. So we chewed on all this information over a little while and came up with, in the end, a design that included a core content of exome and GWAS SNPs, custom content of interest to us. There were some spare positions on the array at this point, and so we asked the the folks on the design team to use that as they thought best to increase genome-wide association coverage. And so this was actually a fairly painless process, over the course of several weeks. Sometimes when people think about these panels, the question always comes up, can you really do a good GWAS with only 200,000 or 300,000 SNPs? And Peter made this point, which is that, with imputation, you can actually do a very good GWAS with numbers of markers on this order. And so what we expect is that over the course of the next year or so, you'll start seeing some very large imputation reference panels, going from the current panels that might have on the order of 1,000 individuals to 10 and 20,000 individuals. And what this little table shows is that as that happens, you expect imputation accuracy to increase for both common variants here with frequency, say, above 1%, but also even more dramatically for rare variants. And so we think that even with these kind of relatively sparse sets of markers genome-wide, we'll be able to have a very good look at the genome of each of these individuals as these panels become available. The genotyping was carried out by Affymetrix as a service. I think, this is really not my area of expertise, but, watching and listening in on calls, I thought the process was relatively painless for sample transfer and sample check-in and QC. As issues came up with samples, there's regular contact with people at Affymetrix, and I say, it looks like this plate, there's a problem with it. We sent a new one back, and there were no major delays. We got the data just at the end of last month, and analyses are ongoing. I'm just going to give you a short preview. Call rates, both per marker and per sample, were really quite good. When we separate markers according to their source, sorry that this is hard to read, whether markers that are already on the core biobank chip or newly added markers to improve GWAS coverage, markers that we had selected but weren't part of the array before, or markers that were newly designed. All of these had very high call rates, above 99.5%, so on average. Association analysis, if you take a set of markers across the genome in regions not previously implicated in psoriasis, the results look quite good. The QQ plot looks nice. If you look genome-wide, you see that the signal is now much more clear than it was before. We haven't had a chance to digest this, but we're really excited about the experiment, and I think the fact that this went from starting to talk about content to getting the results back in about 4 months is really quite kind of a very nice outcome. So I think that's where I am with this, and hope I fulfilled my goal of not telling you things you hadn't heard from Peter and Chris, because those might be wrong, and that's what it is. Thank you.